Our next talk is by Oni Mapp, who's an M13 cohort and currently at the University of Chicago. And Oni is getting her talk set up. And the title of Oni's talk is Analysis of the Role of Prickle 1B during Zebrafish Facial Branchiomotor Neuron Migration.
Good morning, and thank you all for coming to my talk. Today I'm going to tell you about some of the work I'm doing in Victoria Princeton Lab at the University of Chicago. So as you all know, the brain is an incredibly complex organ. It has to be able to process a lot of information in a very small amount of time. And in order for this to happen, the different areas of the brain have to communicate with each other very rapidly. We're interested in the development of the brain, which is an imperative for proper function. And in particular, we're interested in the development of the hindbrain, which is this region here. The hindbrain controls basic functions such as movement, breathing, and heartbeat. During the development of the hindbrain, circuitry must be established that allows the different areas of the brain to communicate. And this often means that neurons have to migrate from one region to another. Our lab is interested in understanding how neurons migrate and the genes and signaling pathways that regulate their development and their organization. So to do these studies, we use the zebrafish, and there are several reasons that we use the zebrafish, some of which are listed here. One of the most important is that they develop similar to humans in that their brains are organized in similar manners, and they have many of the same genes and signaling pathways that regulate development. One important difference is that development is very rapid in the zebrafish. So we start with a one-cell embryo shortly after fertilization. This is the yolk and the single cell emerging from the top. And 48 hours later, we have a fully formed embryo with the eye here, brain here, and the tail. This embryo has a beating heart, and many of the organs are already functioning. Using genetic manipulations, we can do loss of function studies. In other words, examine what happens when a gene is not functioning. And we can make use of fluorescent transgenics. In other words, label a subset of cells with a fluorescent reporter, such as the GFP or RFP, green fluorescent protein. The use of transgenics, in addition to the transparency of the embryo, allows us to follow cells in real time. So as I said, we're interested in the facial neurons. This is a dorsal view of the embryo, in other words, looking down at the back of the head. And as with all the pictures that I'm going to show you, the anterior or the head is to the left, and the posterior or the tail is to the right. This is a GFP transgenic embryo that's expressing green fluorescent protein in a subset of the neurons. And the hindbrain is transiently segmented into regions called rhombomeres, which are listed here 1 through 7. We're interested in the facial neurons, which occupy this position between rhombomeres 4 and 6. And one of the reasons that we're interested in them is that they undergo a very unique migration pattern. They're born at about 16 hours post-fertilization, or HPF, and by 18 hours, they begin to migrate out of rhombomere 4, where they're born, towards the posterior hindbrain. Once they reach rhombomere 6, they stop migrating, so that by 48 hours, all of the neurons reside in this region. So we're interested in identifying some of the genes that regulate this process, and one gene that we've identified is called Prickle 1B. This gene is expressed in the facial neurons as they're migrating, and loss of function of Prickle 1B results in their failure to migrate. So this is illustrated here on the right. This is a wild-type embryo, and this is the GFP transgenic again, and you can see that the neurons are all residing in rhombomere 6 here. When we lose Prickle 1B function, the neurons fail to migrate and are all found in rhombomere 4. Furthermore, through transplantation studies, we've shown that Prickle 1B function is required specifically in the facial neurons for their migration. So the involvement of Prickle 1B in this process is pretty intriguing to us because it's a component of the planar cell polarity pathway, which is a non-canonical Wnt signaling pathway. And normally this pathway is required to organize planes of cells or epithelia. And one prominent example is the back of a mouse. So this is the back of a mouse's neck. And in a normal case, you can see that all of the hairs are pointing in the same direction. And this is a result of the organization of the underlying cells due to planar cell polarity signaling. In a PCP mutant, the underlying cells are disorganized such that the hairs emerge in random directions. However, neurons aren't epithelia. They aren't sheets of cells, and they have their own mechanisms for organizing themselves and polarizing themselves. So the big question is, why is prickle-1b required for facial neuron migration? We think that prickle-1b may be affecting the ability of neurons to extend protrusions. So in a normal case, the cell body of the neuron would begin to extend protrusions in different directions into its environment, looking for migratory cues. Once a direction for migration has been established, a leading protrusion is extended in that direction that allows a neuron to move in that direction. So in the case of prickle-1b loss of function, we think that the neurons either can't extend protrusions at all, or they can't establish a direction for migration and establish a leading protrusion. To study this, I've generated a new transgenic fish that, instead of expressing GFP in the neurons, expresses a cell membrane targeted red fluorescent protein, or RFP. So you can see the facial neurons here, and you can begin to see some of the protrusions being extended. And by following these cells in real time, I've been able to understand what happens in the case of prickle-1b loss of function. So this is a time-lapse movie of a normal embryo during facial neuron migration. This, again, the anterior is to the left. So these are the two rows of facial neurons as they're migrating back. You can see some cells here and here. You can also see the axons exiting from rhombomer 4, where they'll go on to innervate the muscles of the face and throat. I've noted two neurons here, so you can see examples of migrating cells. And when you see the movie, it'll appear as though the whole thing is shifting to the left, but that's because the embryo is growing. So I've marked a stable point with this line. So what you can see is cells such as those marked with these dots moving away from this green line. And this movie is going to loop. So I'd like to point out a few important features of this. First of all, you can see that some of the cells are elongated, such as these cells here in this cluster. And they're elongated in the direction that they're migrating. Secondly, you can see some cells extending protrusions towards the middle. And this is something that had never been noticed before because of the type of analysis that had been done before. This transgenic line that we've generated has been crucial for that observation. So we're trying to understand what these protrusions are extending towards, because we really have no idea. Yeah, I think that's all I wanted to point out. So now what happens in the case of prickle-1b loss of function? So as I said before, the neurons don't migrate to rhombomer 6, they remain in rhombomer 4. And again, I've marked two neurons with yellow dots. However, you'll see one of those dots disappear, and that's because the neurons are migrating out of the plane of the movie towards you guys. So you can see that the cells are elongated in the incorrect axis. Instead of being elongated in the direction that they're supposed to be migrating, they're elongated in a direction perpendicular to that. You can also see some cells extending very short and spiky protrusions. So, for example, I don't know if you guys can see it, but there's a cell here that's extending very short and spiky protrusions, and another cell here, this one marked with the yellow dot. There are also some cells that are extending very long protrusions, and this is something that we also don't see in the normal case. So to summarize what I've told you so far, there are several critical characteristics of facial neuron migration. In the wild-type case, the cells are elongated in the direction that they migrate, and they extend fewer protrusions, including those that are extended towards the midline. Those protrusions seem to be retracted relatively quickly. However, in the case of prickle-1b loss of function, the cells are elongated in the incorrect axis. They extend more short and spiky protrusions, and those protrusions seem to be retracted more slowly. So this suggests to us that the neurons are trying to figure out their environment, but they can't interpret it very well. This also suggests to us that prickle-1b is required to orient neurons and establish a direction for their migration. So one of the future experiments that I'm working on is identifying the underlying causes of these changes in cell behavior. And we think that this may be a misregulation of the cytoskeleton. So the cytoskeleton is the structural support of the cell, And in order to understand this, I'm generating more transgenic fish that will allow us to visualize the cytoskeleton in live cells and carry out similar time-lapse imaging. So this is a double transgenic. You can see the red again, which is the membrane RFP that I've already shown you. And the green is a marker for the microtubule, which is a component of the cytoskeleton. And you can see that in some cells, the microtubules are surrounding or are just inside the membrane or the periphery of the cell. So by carrying out similar studies, we can understand what's going on in the cytoskeleton when we lose PRCL1B function. So I'd like to acknowledge the members of the Prince Lab, especially my advisor, Dr. Vicki Prince, who's in the driver's seat appropriately, and also Monica Rohrschneider, who started this project. And I'd also like to thank the Fish Group at the University of Chicago and the Microscopy Corps for help with imaging. And I'll take any questions that you have.
So have you looked at the expression pattern of PRCL in your...
Yes, so it's expressed in the facial neurons as they're migrating. And one unique thing about PRCL versus some of the other components of the pathway is that it seems to be required within the neurons while they're migrating, whereas other components are required in the environment. So based on some of the traditional models of how the pathway is working, it's not acting in a traditional way. So that's another thing I'm looking at, the interactions with PRCL1B and some of the other components of the pathway.
Okay, so you don't see any asymmetric localization of PRCL?
I'm generating an antibody to do that.
Okay.
We think that there might be. So one of the other components, VANGO2, I'm sure you know, is we think that it's asymmetrically localized within the neurons. And I can't really tell where it is, but it's not everywhere. So when I look at its co-localization with PRCL1B, which I'm assuming I'll see because they're supposed to be interacting partners, hopefully we'll see that it's asymmetrically localized as well and be able to pick out some kind of function from that.
Can you make PRCL1 neuron specific knockouts? Have you done that? Or because you said
So,
you think it just requires neurons.
yeah, so what we do is we do cell transplantation. So we have one embryo that we inject with PRCL1B and transplant those cells.
And just, is there any like PRCL homolog humans that
Yeah.
involved in this?
So as Belinda was saying, many members of the canonical and canonical pathways are required or are overexpressed in colorectal cancers. There are some other applications for it, the closing of the neural tube. So if you lose a member of this pathway, your neural tube doesn't close and your brain develops outside of your skull, which of course is not a good thing, and some other things as well.
So initially you stated that without any good reason that you thought the protrusions might be, the direction of the protrusions might be the most important determinant that planar cell polarity defect might be interfering with. But then later on you showed, I think the most obvious thing, although the fact that the deficient cells lack protrusions, is that their orientation is much different.
Yeah.
So do you think that that could also be
It could also be.
an important factor?
The thing is, and I didn't point it out because it's kind of hard to interpret right now, is that when the cells are born in rhombomer 4, they're initially elongated in that medial-lateral direction, and then they change their elongation perpendicularly to the direction that they're migrating. So we don't see them changing their direction of elongation in the PRCL1B morphin, but it's hard to say whether they don't change their direction because they've lost PRCL1B or the loss of change of direction. It's kind of a chicken and egg thing.
Exactly.
Right.
Is it possible that the mutation could be due to having more than one leading protrusion?
It could be also. So normally what happens is once you decide which direction you want to go, you only extend one leading protrusion. So it would be, it's possible, although I've never heard of any cases where a cell extends more than one leading protrusion. Normally what will happen, it will just extend shorter protrusions and it can't figure out what to do. So it just